Welcome, everybody, to the third BSAN lecture. I'm going to pause briefly before introducing the lecture so we can welcome Arabic speakers and to give them time to switch to the Arabic channel.
Thank you, George. So شكرا جورج ومرحبا بالجميع المتابعون على الزوم والذين يفضلون الاستماع إلى المحاضرة بالترجمة الفورية إلى اللغة العربية. يرجى النقر على أيقونة الترجمة في أسفل الشاشة التي تمثلها الكرة الأرضية واختيار العربية. هذا الخيار غير متاح لمستمعي الفيسبوك. شكرا جزيلا لنمير قسيس الذي يقدم الترجمة الفورية.
Central to our ambition in the BSAN lecture series is to contribute to the liberation of the global cultural community, including the scientific community, from forcible isolation, such as is being imposed on the Palestinian people. Nowhere is such communitarian commitment better illustrated than in Cuba's remarkably successful universal healthcare system. It's universal, not only in the sense that it embraces all 11 million of Cuba's citizens for free, but also in the sense that it relies on and contributes substantially to the global medical community. To an impressive degree, the island has been able to overcome my own country's 6-decade effort to isolate it from the rest of the world, culturally as well as economically. Today's speaker, Dr. Gertrudis Rojas, exemplifies Cuba's communitarian approach to medicine. She's going to tell us about the development of Soberana 1, Soberana 2, and Soberana Plus, 3 of the 5 COVID-19 vaccines developed in the country's biotechnology sector. Independent development has been a necessity since the US and our allies make it exceedingly difficult for our technology, including, for example, the new messenger RNA vaccines, to be imported to the island. Dr. Rojas says the vaccines are non-innovative. She means that they're built in obvious ways on vaccine technology that's already well established in Cuba. That's a strength, not a weakness of the program. Glory and patents are not the point. Mobilizing to protect the nation's people, all of the people, from the worst ravages of the pandemic. That's the goal. Soberana is a team effort, a very large team. In the scientific reports arising from the project, Dr. Rojas's name appears in the middle of long lists of authors. Despite her high position as head of the protein engineering department in the Center of Molecular Immunology in Havana. That's the department responsible for engineering and overseeing large-scale production of the viral antigen, the so-called receptor binding domain, or RBD, at the heart of all 3 Soberana vaccines. As someone myself trained in immunology and protein chemistry, I read the published accounts with admiration for the high quality of the science and for the team's success in coordinating their very diverse contributions. Those long lists of authors also include a number of international scientists who participated in the project, highlighting Cuba's international outlook, an outlook that Dr. Rojas's career itself illustrates and that she values as a way to bring expertise, knowledge, and technologies to her country. She has trained and worked extensively in foreign countries and maintains multiple collaborative networks with foreign scientists. Dr. Rojas's lecture has been pre-recorded. Connectivity in Cuba is just too unreliable for a live presentation. But you can still submit questions during the lecture in English or Arabic. In the chat, just click on the chat icon at the bottom of the Zoom screen, or on Facebook if that's how you are participating today. Dr. Rojas will respond to the questions over a phone connection at the end of the lecture. And now here's her presentation, Soberana Vaccines Against SARS-CoV-2.
Hello. First, let me introduce myself. I'm a biochemist from the Center of Molecular Immunology in La Habana, Cuba. And today, I would like to share with you my personal experience in the process of developing Cuban vaccines against COVID-19. My participation in this process was just a very small chapter in the whole story, but I think it could be interesting for you. Now I will talk a bit about my country. Cuba is a very small island in the Caribbean Sea, in the Central America area, and we are very, very close to the United States. At least in geographical terms, we are very close. We are over 11 million of people here, and La Habana, my city, is the capital city. Cuba is usually known by the sunny beach Varadero, by some products like tobacco, cigars, sugarcane, rum, but Cuba is also known because of a very early development of biotech industry. During the 80s and 90s, several research production institutes were created, and by that time the biotech development was starting in the whole world. So we incorporated really early to this process. The first center was the Center of Genetic Engineering and Biotechnology, but it was quickly followed by an institute devoted to vaccines, the Feinlein Institute, the Immunoscience Center, devoted to diagnosis, Biocen, the Center of Molecular Immunology, the place where I'm working right now, and other institutions. This development didn't stop. And in 2012, the group Biocupa Pharma was created. Biocupa Pharma is a group that coordinates the activities of more than 30 biotechnological and pharmaceutical enterprises, including joint ventures in Cuban territory and abroad. Within this complex biotech system, the Center of Molecular Immunology is quite important, and this is the center. And here I have my lab, the Protein Engineering Lab. Here you can see the lab with some of the members of my group working here. If I want to define the Protein Engineering Department, I would say that this is the place where new proteins are born. We use large molecular libraries, large collection of molecules, to discover and optimize immune-related molecules to perform the desired functions. And by using this kind of technology, we can develop anti-cancer antibodies that attack cancer cells, but we can also modify a group of immune modulators that mediate cell-to-cell communication and are very important in regulating immune responses. They are called cytokines in general. Once we have the ideal protein, once we have optimized the protein for a given function, we are able to do high throughput production of multiple proteins at laboratory scale to study their activity in vivo, in vitro, to know exactly what functions do they do. And once we have the protein we were looking for, we can then generate stable cell lines that produce these therapeutic proteins, And these cells are like protein factories, suitable to go until the industrial scale. So we are ready to go from the initial experimental research in the lab to the production unit to obtain molecules to treat patients. What happened after COVID-19 emergence? Since 2020, Cuban scientists were preparing the scenario for treating and preventing COVID-19 at both public health institutions and also biotechnology institutions. In March 2020, the first cases of COVID-19 were detected in Cuba. A few days later, my lab was incorporated to the vaccine development project. This was a great opportunity for us, actually. In May 2020, something important happened. The Cuban president organized a meeting with scientists from the biotechnology field to confirm the decision of making our own vaccines. By that time, we had already realized that the availability of vaccines against COVID-19 could be limiting. But even if there was a large production of COVID-19 vaccines, our resources to buy them would be very, very, very limited, because we are a poor country and because the pandemic was aggravating our economical situation. So the only chance for us to vaccinate Cuban population was to be able to develop our own vaccines. And a bit more than 1 year after this decision, in July 21, our regulatory authorities authorized the use of emergency of the first anti-COVID-19 vaccine in Latin America, developed in Latin America. The name of this vaccine is Abdala, and it was produced by the Center of Genetic Engineering and Biotechnology. Just a few days later, in August 2021, Soberana vaccines also received the authorization to be used. If we are gonna talk about the vaccines, I have to start defining what is a vaccine made of. And a vaccine always have two kinds of components. Non-specific components are those that enhance the immune response against the vaccine, or even enhance the global immune response of the vaccinated individual. There are multiple compounds of different chemical nature, biologic, organic, inorganic, that can be included in diverse vaccines to improve the responses. But it's very important to have specific components, also called vaccine antigens. And vaccine antigens are particular pathogens, or parts of the pathogen, or compounds that resemble the pathogen. The response against these specific components is the one that attacks the target pathogen, protecting vaccinated individuals from getting the disease. Now I would like to mention a few examples of specific components in the case of antiviral vaccines. We can use as specific components the whole virus, attenuated or inactivated to make it harmless, of course. Or we could use modified non-pathogenic viruses that contain molecules that simulate the original target virus. So these are chimeric viruses simulating the original one, the one we want to combat. But we could also use synthetic compounds obtained by chemical synthesis, which resemble parts of the virus. A very simple way to obtain vaccine antigens is to produce viral proteins or protein fragments through genetic engineering, and this is the way we are using in our vaccines. I'll talk a bit more about this variant later. We could also use nucleic acids coding for the synthesis of viral proteins to induce the synthesis once the vaccine is in the body. So in these cases, we do not inject with the viral protein. We inject with the DNA, and the viral protein is produced inside the body. The Cuban strategy to produce anti-COVID-19 vaccines had three three main principles. It involved several institutions and multiple production facilities. We were looking for redundancy as a way to obtain a robust system. So if one production facility failed to produce a vaccine component, this wouldn't be a terrible problem because other production facilities could compensate for that. Our strategy also included the development of several vaccine candidates, which together could cover the whole Cuban population and eventually could be combined to achieve better results by obtaining mixed vaccination schedules with several vaccine candidates, later vaccines, once they were approved. And our strategy took advantage of already established technological platforms to accelerate the development of the vaccines. We were not very innovative in terms of the technological platforms we tried to produce vaccines very similar to the ones where we were already using and producing at the country. Our vaccine antigens were produced by genetic engineering. The idea behind genetic engineering is very simple. You take a gene, a piece of DNA, coding for a protein of interest, we introduce the gene into living host cells, and then you convert these host cells into protein factories. So the cells start to produce at a very high level the protein coded by the introduced DNA, and you can obtain huge amounts of the protein in a quick way. In the case of Cuban vaccines, RBD is the specific component. The protein RBD is the specific component. To explain what is RBD, I'm showing here a representation of SARS coronavirus 2 virus. This is the virus, and the virus has these spikes that are similar to the ones in a crown. That is why the virus is called corona. But if you look in more detail in the structure of each spike, it's actually a trimer of 3 different spike proteins highlighted here in different colors. So the structure is very complex. But within this complex structure, you can identify, highlighted here in yellow, the RBD. RBD means receptor binding domain. And the function of RBD is binding to a receptor on target cells. So receptor binding domain, here in yellow, binds to the receptor on the cells, and this is the first step of virus infection of the cells. So if you interrupt this process here, if you disrupt binding of RBD to the receptor, you are preventing infection. And that's what vaccines usually do. Vaccination with the RBD induces antibodies that recognize RBD here, different sites of RBD, and these antibodies are able to block binding to receptor on these cells. So infection is prevented in that way. RBD is a complex protein. RBD is represented here as a sequence of more than 200 building blocks. All proteins are composed by building blocks that are called amino acids, and RBD has more than 200 of them. Each letter represents a different amino acid, and here the letter C is always underlined. Why? Because cysteine, C means cysteine, and cysteine is a very special amino acid with a highly reactive sulfhydryl group, SH. And as this group is so reactive, 2 cysteines can form a disulfide bond, represented here as SS. And such disulfide bonds connect distant building blocks and are essential to shape the 3-dimensional structure. So this cysteine here could bind to this cysteine here, or to this other cysteine here, and the network of disulfide bonds that have to be exactly paired is one of the essential factors that maintain the three-dimensional structure of the protein. So to obtain this structure, you have to form correctly the 4 disulfide bonds involving 8 cysteines. 1, 2, 3, 4 bonds here. And we saw that both yeast and mammalian cells were able to produce properly folded RBD resemble the protein fragment in the virus. So RBD obtained by genetic engineering in yeast or mammalian cells was able to reproduce the structure of the RBD in the viral crown. The use of yeast cells as host cells to produce viral RBD gave rise to 2 of our vaccine candidates, Mambisa and Abdala. Abdala is already an approved vaccine for human use. It was the first to receive the authorization. And these 2 vaccine candidates were developed by the Center of Genetic Engineering and Biotechnology. But we also used mammalian cells in Cuba to produce viral RBD. And Soberana vaccine candidates produced by the Finlay Vaccine Institute made use of the RBD produced by the Center of Molecular Immunology, and this development process started in my lab. Now, if we are going to talk about Soberana vaccines, it's good to say that Soberana means sovereign. So the idea behind the development of this family of vaccines was to be independent in terms of obtaining the vaccines. Here you can see three different formulations of Soberana vaccines. But producing the vaccines required to make the Soberana vaccines was very, very challenging. It was technically difficult. Why? Because our colleagues from the Finlay Institute asked us to include an additional cysteine, which was non-paired. It was an alone cysteine. We could say it in that way. At position 538 of the spike protein. So, and this cysteine was very important for vaccine design, as you will see. But the presence of an additional cysteine, now they are 9, with highly reactive sulfhydryl groups, complicated the folding process, complicated the biosynthesis process of the protein to obtain the correct structure. And also the Finlay Institute asked us to achieve the simultaneous production of 2 different molecules derived from RBD in a single biotechnological process. And if making a bioprocess to obtain one protein is complicated, making a bioprocess to obtain 2 different proteins simultaneously, of course, was even more difficult. But we decided to try, because it was really important to obtain both products. This is the product the Finlay Institute asked us to produce. It's an RBD similar to the one I was talking about, but with this additional cysteine. And here in the structure you can see this additional cysteine here in yellow, and it was important to avoid that this cysteine had the possibility to make bonds with the other cysteines, because they were committed to form their own disulfide bonds to keep this structure. So this cysteine should stay apart from the others. And what should we do with the highly reactive sulfhydryl group during the biosynthesis process in mammalian cells? Well, one of the molecules that we had to produce was a dimer. to form this dimer, two cysteine residues from two different molecules had to bind. And in this case, we could obtain a dimer of two RBDs bound here by another disulfide bond. And this dimeric structure could be good to induce a very potent immune response. But this was one vaccine formulation. But for the other vaccine formulation, it was important to preserve the sulfhydryl group free to be conjugated to a second highly immunogenic carrier protein, tetanus toxoid. So the idea here in this second vaccine formulation was to have tetanus toxoid, which is a very common vaccine against tetanus. But if we conjugate other compounds to this protein, these other proteins or compounds in general could also become very immunogenic, because the complex as a whole is very immunogenic, so it induces a very strong response. So this reactive group was very important to do the conjugation. So the idea at the end was the following: to obtain part of the molecule as a dimer and to obtain part of the molecule as a sulfhydryl free version to be conjugated to the tetanus toxoid. At the end, we obtained dozens of different cell lines producing a mixture of monomeric and dimeric RBD. These cells came from hamster ovary, CHO cells that are very common in industrial biotech facilities, and our cells were able to produce both forms of the antigen. Here you can see, I will not go into deep technical details, but this method, an electrophoresis, is able to show the size of the produced proteins. And you can see that in every case, each line is a different cell line here. And all of these cell lines produced two main forms of the antigen. A big one that is a dimer, and a short one that is a monomer ready to be conjugated with tetanus toxoid. This is the result of our production at a laboratory scale. And if we break the disulfide bonds before doing the technique, then all the protein becomes monomeric, confirming that the dimer is formed because of a disulfide bond between two different molecules. So this was a very successful result to be able to obtain cell lines producing both versions of the vaccine antigen. And this is my team. This team made this part of the work to get the suitable cell lines. The best producer cell line at the end was transferred to industrial bioreactors. So we stopped growing the cells in this kind of a small flask at the lab, and then the cells went to very big bioreactors up to 2,000 liters, and in these industrial facilities the cells are used to produce both vaccine antigens, which is something very amazing for us to see our cells going here to the industry and going to the real life application. All Soberana formulations we produce have been useful at the end. 2 doses of Soberana 2, that is the conjugate between RBD and tetanus toxoid, are being used to induce immunity in children and adults, followed by a third dose that is just a recall dose to enhance the already existing immune response, and this recall dose is given with Soberana Plus, that is dimeric RBD now without the tetanus toxoid. This combination was very good. First, tetanus toxoid helped to prime the response, but dimeric RBD was enough to potentiate the existing response. Here I'm showing some images because Cuba was the first country to introduce massive children vaccination, and Soberana vaccines were crucial for that goal. Then a single dose of Soberana Plus, which is the dimeric RBD, has been very effective in boosting the response of convalescent people. Here the priming doses were given by the virus in infected people, but then it's enough to give a single dose of this Soberana Plus vaccine to obtain very, very high immune responses that prevent future infections with the virus. And finally, we have another formulation, Soberana 1. Soberana 1 is dimeric RBD, but in this case is combined with membrane vesicles from Neisseria meningitidis. Neisseria meningitidis is a pathogen. It's a bacterial pathogen that causes meningococcal disease. But these bacterial components are very, very strong in stimulating what is called the innate immune system, so the first line of defense against pathogens. And the combination of a specific vaccine antigen, in this case our dimeric RBD, with this non-specific immune stimulator is being studied now for booster doses to keep the immune level very high after the initial vaccination. So this formulation is also being studied for a particular application. Now I will show you the Cuban vaccination in global numbers. These are the data from April 2022. We have almost 11 million of persons that have received at least one dose. More than 9 million have received 2 doses, and most of them have also received the third dose. In total, almost 10 million of persons have completed the immunization schedule, and this represents almost 90% of Cuban population. So the coverage is really good. And now more than 6 million of people have received additional boosters after several months of the initial vaccination schedule, to maintain their immunogenicity levels very high and to keep protection. And that's how we are fighting Omicron, the new COVID-19 viral variant, with additional boosters to keep the immunity of Cuban people at very high levels. Now I'd like to comment what did we learn from the Cuban battle against COVID-19. The first lesson is that it was possible, although quite difficult. This process was not easy at all, but it was possible to control the disease despite the negative impact of our own economical problems and of the external pressures dictated by the blockade of the United States of America against Cuba. But it was possible to control the disease. We had very bad moments. We have a sanitary crisis in July, August 2021, but now the disease is under control. Then our vaccines entered the stage of massive application, and we were able to control the situation, and the evolution now is nice, actually. We were able to get preventive and therapeutic agents to combat the disease because we were using mainly products developed at Cuban labs and produced by our biotech industry. And this was possible because scientists and facilities were ready when the disease came. So we are now seeing the results of many decades of previous investment and previous study. So this is the real reason why we were ready to combat the disease now. The organization of our public and universal healthcare system was critical to grant access to these products, including a very fast vaccination campaign. Because in July or August, when we were ready to apply our vaccines after finishing the clinical trials and so on, it was to be organized a huge campaign to vaccinate everybody in a very short time, and this was possible because of the work of our medical doctors, our family doctors, and the progress of this vaccination campaign was really impressive, even for us. The Cuban strategy of communication was effective to promote people understanding and support of vaccination and other sanitary measures. We didn't have serious problems of vaccine rejection, of anti-vaccine movements, because Cuban people in general was aware of the importance of vaccination, and most people understood why should we do it. Finally, our goal of being independent did not mean at all that we wanted to be isolated. We got help from foreign scientists from the very beginning of the process. We got help from other governments and from solidarity groups in general, and this help was greatly appreciated and made a real contribution. When we were trying to vaccinate Cubans, we received a lot of donations of syringes, for instance, and this was really important to be able to convert our vaccines in a practical success. You could see in this slide is about politics, but it's not exactly about politics. Politics is related to everything, but this slide is just the summary of how a very poor country could control a disease that has been challenging for the whole world. We did not do it in a perfect way. We did not do it better than the other countries, but we were able to do it in a very difficult situation. And this is something that other people, other countries could have as a promising precedent. Finally, I would like to say thank you first to all the teams involved in the development of Cuban vaccines. There were many scientists and many people in general at the Finlay Institute, at CIGB, here at CIM, in Biocen, and also in our public health system. And the vaccination would not have been possible without the coordinated efforts of all these people. I also want to thank the organizers of these lectures for giving me the opportunity to share my story here. And of course, I want to thank you for your attention. Thank you.
Okay, my name is Marco. I'm part of Scientists for Palestine. I am taking it from here for the Q&A session because, as George announced before, it's been rather challenging. So I will be communicating with Dr. Rojas by phone. She's already actually here online. So let me start by thanking her for this great lecture. It's it's in my experience, it's very hard to make science understandable without losing its depth and its complexity. So this was really impressive. And it's even more rare to have a speaker and have a lecture, which at the same time has technical content and lesson for society as a whole and politics. So thank you very much for this. This is certainly one of the highlights of this series. And so, okay, if there were many questions I saw in the chat, of course. So if someone, Bernardo, probably can share the slides, we can start asking them to Gertrudis. We should be able to answer live. I should also mention that, yeah, so we're using WhatsApp. So I apologize. I hope the connection will hold and it will be good. I really would like to mention the fact that so Zoom is blocked in Cuba because of Zoom's will or the embargo, and this didn't make it any easier. Even for Dr. Rojas already to be in the Zoom room was challenging. So without further ado, let's go with the first question. So Dr. Rojas, which other COVID vaccines, Moderna or Pfizer, use genetic engineering?
Thank you, Marco. Can I answer already?
I think so. Yes, people are nodding, so they can hear you.
Yes, actually, many vaccines are based on genetic engineering, but different modalities of genetic engineering. For instance, mRNA vaccines like Pfizer or Moderna make use of preparing a nucleic acid that is going to be injected into the body of the person, and then the genetic engineering will take place in the body of the person. So the nucleic acid will dictate the synthesis of the viral protein there inside the body, and then the body will respond. What we do is something related in some extent, but a bit different, because we didn't inject the new genetic material into the body of the person. We introduced this genetic material into cells in a factory, in industrial bioreactors. So our genetic engineering is outside of the body, but all of them are genetic engineering in some way. Other vaccines make use of genetically modified viruses. So they make the genetic engineering of the virus outside of the body, and then they inject the virus, and the virus introduces the new genetic material into the cells of the body. So it's again a kind of genetic engineering that takes place, at least in the final steps, inside the body. So our genetic engineering is outside the body, but the general principles are related, of course, are technologically related. But yeah, genetic engineering is on the basis of many vaccines in general. Genetic engineering is in the basis of the whole biotech industry in general.
Okay, thank you very much. I think the next question is actually a follow-up to this. So yeah, so since there are various ways of doing genetic engineering, what are the arguments to choose one of the possible types of vaccine? Attenuated virus, synthetic proteins, mRNA, etc.
I would say that the best vaccine is the vaccine you are able to produce with good quality. So a very important element, not the only one, but a very important one, is which are the facilities you have to produce your vaccine, which is your experience, the expertise of your scientists, and so on. So there are excellent technical and scientific explanations for the success of one vaccine or another. But the most important thing is the one that you know how to produce. The know-how is the most important thing in this problem. Having said that, I would say that you also have to perhaps to try one or two or several alternatives in order to obtain high immune responses. So for your vaccine, you need to be able to produce it, but you also need a very high immune response when you apply this to the people. So in this case, if you compare several vaccines or several modalities of a given vaccine, you can obtain the best results. For instance, we did a combination of more than one antigen during the vaccination schedule, and this was successful. But the ability to produce is really crucial.
Thank you. And actually, before we move on, since you mentioned facilities, I think there is probably a related question later, but I'd like to ask you directly, how was what was the effect of the embargo on availability, on how easy or hard it is to produce, say, vaccines, say, in particular, buying or producing equipment and quality of the facilities?
Well, one of the problems with the embargo, there are many problems, but one of them is that our ability to buy supplies is very limited. Even if we have the money, we cannot buy to a company that is US based. From American companies, and also from companies that are not exactly American, but have a close relationship with American companies. So they cannot sell anything to us. This is the first problem. So we have to look for another alternative. So the spectrum of companies that we can negotiate with is quite limited. And also, if we really need something that is produced by an American company, the only way for us is to go through intermediate companies. And this can be a long process, and it's more expensive, of course. So our ability to receive materials, even for research, it's the same for research materials or for industrial supplies, and for medicines, it's more or less the same.
Okay, thank you. So we can maybe move on to the next question. Have Western countries requested to test and approve Cuban vaccines? Are these vaccines available and being used internationally?
Yeah, the vaccines have been available for the countries that have been interested in using them. For instance, Venezuela, Nicaragua, Iran, Syria, and there are now some agreements with Italy to organize clinical trials in Italy.
But
these are very recent agreements, as far as I know. And Italy is perhaps the first European country that will test our vaccines.
I'm actually happy to hear that, since I'm Italian myself. Okay, so maybe we can move on. What earlier vaccines did your lab engineer?
Sorry?
Which earlier vaccines, which other vaccines did your laboratory engineer? If any. Can you hear me, Dr. Rogers? Not well. Okay, so let me try again. Did your lab engineer other vaccines earlier on, or other proteins for other vaccines? And if so, which ones?
No, actually, well, my lab in particular, we were not devoted to vaccines. We were involved in producing antibodies, anti-cancer antibodies, and also cytokine variants. For instance, we are working a lot with the modification of interleukin-2, a molecule central in the immune responses, to make it more effective against cancer. But my lab in particular was not involved in vaccine development, but we were involved in production of proteins for therapeutic use. So that's why our expertise was able to contribute to this vaccine project. But we were not vaccinologists. We are not vaccinologists.
I see, okay. And so, yeah, actually you mentioned that this was a great opportunity for your lab. How hard was it then, I would like to ask, to then start producing vaccines for you? Was it a big change, or did you already have everything in place?
Actually, we had experience. That's why we were called to join this effort. But it was a great opportunity, mainly because all the scientists, not only us, all the scientists by that time were trying to figure out how to contribute to the fight against COVID-19. I think this was the dream of many scientists in many countries. And for us, the opportunity came with this group, with this family of vaccines. And we were able to keep our lab working. This was also something very important for us, because at the beginning, when the sanitary measures were quite strict, many other institutions had to stop. And we were lucky, we could continue doing what we like to do, but also we were contributing to save the nation somehow. So that's why it was a great opportunity. We were very happy with our previous project. We will continue, or we are starting to continue all these projects as well, but this opportunity was special to do something important to combat an important problem.
Okay, thank you. Yeah, it's really great to hear scientists that actually are excited about doing something for people. As a theoretical physicist myself, that's not always what drives us, but it's amazing that there are people who are driven by that. So next question, please. Does the structure of RBD change between virus variants?
Are you talking about new virus variants?
I think it's in general. So if you take, say, 2 of the variants of COVID-19, does the structure of RBD change between them? I think we might also ask if you can expect the same vaccine to be effective against multiple variants.
Yes, yes, there are structural changes between the viral variants, of course. That's why my lab has also been in charge of producing the new RBDs, the modified RBDs. And with these RBDs, we were ready to produce a new vaccine if necessary, but we were also able to show that the vaccines we had already were able to inhibit the binding of these new RBDs to the viral receptor. And there are also other studies, of course. There are studies with virus in other institutions in Cuba, and the sera of vaccinated patients was in general able to neutralize the viral activity of these new viruses, the modified viruses that emerge during pandemics. So at least until now, we have seen good promising responses to our vaccines regarding the new variants of the virus. But if we have to modify the vaccine, we are ready to do it.
Okay, thank you. So next question, please. Why did the Finlay group ask for that annoying extra cysteine?
Yes, yes, because they have a lot of experience in chemistry. So they know very well how to do conjugated vaccines. So they were trying to make a conjugate vaccine because they have a lot of experience and successful results. So they want to make a conjugated vaccine. And this cysteine was totally necessary to make the conjugation. It's a good way to conjugate the RBD to the tetanus toxoid. So it was difficult for us to have the cysteine there, but it was very good for them and for the final story of the vaccine.
So for somebody who does not understand anything about this, conjugation means attaching. So it was necessary to attach the protein to the tetanus toxoplasmon. Is that correct?
Sorry?
I'm just asking for a clarification.
Sorry?
You said various times, you mentioned the word conjugating. So I was just trying to make sure that I understand. Is conjugating is just means attaching?
Is to establish an stable bond between the carrier protein, the other protein, in this case, tetanus toxoid, and the protein, the viral protein, in this case, RBD.
Thank you.
Is to make a whole complex with two or more different molecules.
Thank you. Okay, so next question, please.
So you can inject the whole complex.
Thanks. Okay, so the next question is, how can you tell if the cysteines are properly paired with one another?
Can you repeat?
Yes. How can you tell if the cysteines are properly paired with one another? So if the structure of the protein is correct.
Are you saying that if we know that the cysteines are properly paired?
Yes, that's the question exactly.
Yes. We know that because strictly we know that because of mass spectrometry studies. In mass spectrometry studies, you can see where the cysteines are and which bonds are forming between them. This is something you can check directly. But also we know that the structure in general, the global structure, is the correct one because this recombinant, these genetically produced RBDs are able to bind the receptor and are recognized by the antibodies in the sera of people that have been infected by the virus. So this recombinant protein, this artificial protein, resembles the one that is in the virus and is able to bind the same receptor than the natural protein. So we know that the structure is correct in general. And we know specifically about the disulfide bonds because mass spectrometry studies told us this directly. We have a very, very good mass spectrometry group at CIGB, at another institute, and they confirmed that our RBD is a very good one.
Okay, thank you. We have time maybe for a couple more questions. One of the vaccines you mentioned is Abdala. How does a Cuban vaccine come to have an Arabic name?
Yeah, no, the reason is very simple. Our national hero, Jose Marti, was also a writer. And 2 centuries ago or something like that, a bit less actually, but a long time ago, he wrote a poem about a young man in Nubia who was defending his homeland. And this example, although it took place in a very far place, was very inspiring for our national hero. So his poem was named Abdala, and then the vaccine took this name to honor our national hero.
Okay, thank you. So now with this question, besides science and politics, we also have literature in this seminar. So it's really about the unity of knowledge. Okay, so another question. Is it true that many individuals from outside Cuba went to Cuba to get vaccinated?
Well, I don't have all the information, perhaps, because I was involved in the laboratory steps, not in this massive application of the vaccine. But at least I know the example of a clinical trial that involved Italian citizens, but here in Cuba, not in Italy.
I see.
Perhaps there are other examples, but I don't have the precise information actually.
I see, thanks. So yeah. Okay, so one more question, I think. What was the role of the CDRs in the vaccination campaign? Of the CDRs, which I actually am, I don't know what are the CDRs?
CDRs.
Yeah, CDR.
Are you talking about antibodies?
Possibly, yeah. But that's how it was asked.
I mean the question. CDRs, I meant the Comitato della Rivoluzione.
Ah, sorry, Mario. Okay, thank you.
Comité
this was
de
not a-
vencer,
this was-
defender la revolución.
Oh,
yeah. So it was antibodies, but not
Yeah,
not for the virus. Yeah.
yeah. You are talking about the other kind of CDRs, the committees to defend the revolution.
Yeah. So
Is
that
that
was the
okay?
question. Yes, exactly.
Yeah.
Thank you.
Well,
Mario.
actually, the CDRs became important in this vaccination campaign because the president of the CDR, that is at the end a communitarian organization. So the president of the CDR was many times in charge of reminding people that they were scheduled to be vaccinated today or tomorrow or any day. For instance, in my CDR, in the region where I live, the president of the CDR was very active trying to organize this vaccination campaign that was huge. As a campaign, it was huge. But in every small place of the country, it involved simple people like these presidents of the CDRs that were in charge of organizing everything, of making sure that everybody remembered the vaccination. So they were important, actually.
Okay, thank you very much. I think given that it's already more than an hour into the seminar, I would say that we can stop here. Thank you for answering all of our questions. This was really great. I'll give back the word to George, who is going to conclude. Thank you very much, Gertrudis.
Okay. Okay, thank you. Thank you for your attention.
Well, yes, people are clapping. I don't know if you can hear us clapping, Gertrudis, but this is a most inspiring lecture, and many people in the chat have commented on what an inspiration the attitude and the success and the science is for all of us. So we are very, very grateful that you were willing to do this for the BSAN lecture series. I thank everyone in the audience. There have been some great questions, including questions that are impressive to me as a biochemist, that very similar field to Gertrudis herself. So we're going to say goodbye until the next BSAN lecture, which will be in September. And here is the announcement of it. So Salim Tamari from Birzeit University will be giving the third BSAN lecture in September. So that's September 14th at the same usual time. And until then, goodbye.